Welcome to your third of 4 fully vodcasted lectures. If you're watching this in the lecture hall, then it is Wednesday and it is March the 6th. And of course our homework was due yesterday, so we find ourselves looking into the next week and homework number 6 that'll be due on Tuesday. So just plugging and chugging along with each one of those Tuesday due dates. We do have the practical exam coming up tomorrow, and I feel like I've announced that so many times in lecture now that you're probably starting to get bored of it. I will just briefly mention that, of course, we do have those skill sets, the focusing of the microscope, the aseptic pipetting, and the T-streaking, and that there is a review posted under additional information on the lab website, as well as the review that maybe you went through with your TAs in lab. So it should be totally fun, totally good, and I hope you all rock the socks off of that exam. So one other thing is that if you're still working on finalizing your hypotheses, keep those rolling and keep those coming so that we can make sure we have something that's very finalized on record for your poster presentations. With that being said, we should go ahead and jump in. We left off a little bit abruptly last time in the middle of the action of replication, and we want to follow up now, beginning with what happens at the lagging strand and how does that get synthesized discontinuously. The first thing that happens on the lagging strand is that an enzyme called primase gets involved. Well, primase is actually an RNA polymerase, meaning that it actually makes RNA instead of DNA. Well, you're thinking, what is the purpose of that? I mean, why get an RNA polymerase involved? Aren't we just trying to duplicate DNA? Well, the trouble is that, remember, DNA polymerase with all of its neediness, well, it needs a primer, right? And it can't start cold. But on the other hand, RNA polymerases can. So RNA polymerases can start cold and create a little tiny primer that then from there DNA polymerase can build on. So this is what happens. RNA polymerase comes in and synthesizes a little tiny primer. That's what is being shown right here in the red. These are little tiny RNA primers. And so this is the little 5' end of that little primer. This is the little 3' end of that primer. And so it leaves an open 3' hydroxyl from which the lagging strand can begin to build DNA. So it gives a primer for DNA synthesis. So now DNA polymerase can come in and make little fragments of DNA. Now remember that this strand is getting looped back into the replosome, so it's actually having to loop around. So what happens actually is that these little primers loop back in so that the 3' hydroxyl is set up to go in the direction of the replication fork, and a little teeny piece of DNA is made. So it's like, it kind of goes like this, you know, so here's our replosome going along, and here's our lagging strand, and it loops around and it goes zip, and it loops around and it goes zoop, it goes like, yeah, just like that. So discontinuously is how that happens. So I don't know, maybe I can show that better, even just like thinking about it this way. It's heading this way, and the lagging strand loops around, and it's like these little pieces go like this. And I'm going to show you some awesome videos, and I have an awesome model for visualizing this. But it's short little spurts of synthesis of DNA in order to allow this lagging strand to be made. So RNA primers are made first, and then by looping back around, we get the synthesis of complementary DNA by DNA polymerase 3. And then we end up with the little Okazaki fragments is the name that's been giving them. Yeah, of course, based on the name of the discoverer. And so these little Okazaki fragments form, and so DNA gets put in, starting from the open 3' hydroxyl of the primer, and then a little piece of DNA is made until it hits the butt end of the next primer. So this just keeps on happening as that lagging strand is made. And the little Okazakis are made until they reach the initiating point of the next fragment. Then we have a different DNA polymerase that gets its head involved. It's going to remove the RNA primers, take those out, and replace them with DNA. This is called DNA polymerase 1. So if you want to write that down, you can. That this is a different DNA polymerase, and we'll call it's called DNA pol 1. So we can write that down. DNA pol 1. So another important enzyme. So it removes those RNA primers, replaces them with deoxynucleotides to create what looks like a pretty confluent strand of DNA. However, it leaves still regions where there's a nick that needs to be sealed. And here you go, your favorite enzyme, Eric, ligase, that seals those nicks. So in the end, we end up with something that looks a lot like, because see what happens, of course, is that DNA polymerase I removes these primers, right? It takes them out and removes them. And DNA ligase comes in and seals the nicks here. So we end up with something that looks like it was synthesized 3' to 5'. Wowzers. I've got a lot of models and a lot of visuals to show you this, because in fact I find it was really hard to understand until I was able to build my own model and to watch some of these videos online. So that's where we're going next. As we continue our process of trying to visualize this replication fork and what's going on the lagging strand of DNA, let's go ahead and draw it out to begin, and then we'll go from there and we'll look at a model, some pictures, and we'll also look at some awesome animations that are available online to allow you to better visualize the whole process. So I'm just going to jot down what we've been looking at, where we have the replication bubble and our 5' to 3' strand, and then of course our 3' to 5' strand. And we can draw leading and lagging strands on really both sides here, recognizing that this strand would be synthesized continuously, 5' to 3', and this strand would be synthesized by the other replosome continuously, 5' to 3'. But of course these would be our lagging strands that I haven't yet drawn in. So let's go ahead and take a quick look at how we might go about drawing those lagging strands, and recognizing that the first thing that would happen is that primase would lie down these little tiny primers. So these short RNA primers would be the first thing to be set down in spurts, of course, because as the replosome is heading one way, it can kind of synthesize these short bursts of RNA primers, where they're synthesized in the 5' to 3' direction as always, but it looks like they're kind of going backwards because they can be short bursts that allow that to take place. So recognize this then as the 3' end of this short primer, and this as the 5' end. And the same would be true, that would be the 3', 5'. I'll label it on this one because we kind of get scrunched on room. 3' and 5'. And the same thing is true on this other lagging strand, where we would have these little short RNA primers again, with this being the 3' end, the 5' end, and so on forth. So now, in order to build on this 3' hydroxyl that we see right here, we would have to get the looping back around of this strand, this lagging strand of DNA. So we'll actually have to loop back around and insert into the replosome so that building can take place beginning from the 3' hydroxyl. So DNA will get synthesized, and I'm just going to show it in green here, from that open 3' hydroxyl in this direction, until it hits the butt end of the next primer. And the same thing would be true here, from this end, this 3' end of this primer, until it hits the butt end of the next Okazaki fragment. So these are making those Okazaki fragments, that sort of discontinuous kind of spurt-filled production of DNA. So the same thing would happen here, from the 3' hydroxyl until it hits the next primer. And of course then we know is when DNA polymerase I gets involved with the process, and it comes in and it actually gets rid of these little primers that are made of RNA. So it actually removes those primers, and we see that they can be filled in with DNA. So it's a little hard to erase though. So these will get all filled in with DNA, and a little easier to show down here that will essentially all get filled in. And of course we know ligase will seal the nicks that exist there. So eventually we end up with something that looks like it was in fact synthesized 3' to 5'. So this one was a little less well formed in that drawing, but essentially the same thing would happen there, that it would get filled in and ligase would seal the nicks. And that is the discontinuous synthesis of DNA on the lagging strand. But if that picture doesn't do it for you, it just doesn't help you understand, then I'm going to go and look at a couple of other pictures, some other images, and also some animations. A picture that comes from a different version of the textbook, but I think it's somewhat helpful in recognizing that here is DNA gyrase, helicase, single-strand binding protein, or SSB, is what binds to the single-stranded DNA, keeping it single-stranded so that it doesn't try to pair off with anything else in the interim process in which new DNA is synthesized. So what happens with the leading strand, of course, is that it's just continuously synthesized, but on the lagging strand, notice here are the RNA primers. And then notice how it loops back, it loops that lagging strand back, and then can synthesize from the open 3' hydroxyl, which would be right here. It can synthesize, and then when it hits the end of the next one, it drops it, and then picks up from the next primer. So that's how that would work as the fork movement was headed in that direction. So that may be another visual that might be really helpful. But everybody's got to love this next one. First thing that we see in this animation is just simply the process of unwinding DNA. So I thought a little action on unzipping genes was in order. And then the next video that we'll look at actually literally shows the replication fork and the leading and lagging strand synthesis. So all of this happens pretty quickly, so keep up with the changes and hopefully try to look in all places at once so that you can see all of the things that are happening on the replication fork. Okay, here we go. Here's the synthesis of the leading strand down here. This is the lagging strand. Notice that it is synthesized in an Okazaki, has dropped it, hit the end of the next primer, picked it up again from the 3' hydroxyl of the first primer, hits another Okazaki, drops it, and pulls it back in again. So this is what's going on the lagging strand, while the leading strand just simply getting synthesized continuously. So this is a pretty sweet animation. You can find it online. It's the Walter and Eliza Hall Institute, and you can look that up and look for the replication animation, and you will definitely find that very easily by just simply Googling this. So this is a sweet way to visualize, but I think maybe I really didn't understand replication until I had built myself a model. So I'm going to bring that model up and we can see what this does for us in terms of visualizing the replication fork. So recognize that this is the original DNA, whether it would be plasmid or chromosomal. Now I'm not showing the whole circle, I kind of cut it off just for visualization purposes. But what we see here, this is the replisome, so this is representing one replisome. This likewise is representing one replication fork. So notice that on this replication fork, the way I have it labeled, this is the leading strand right there. So the new purple DNA, that's the leading strand, getting synthesized continuously as this replisome heads this way down the fork. But here is the lagging strand. And what we can see on this lagging strand is that this fluorescent green is the primer. And on the little RNA primer, you can recognize the 5' end and the 3' end. And know that we have to build from that 3' hydroxyl. So from that 3' hydroxyl, this will have to loop around. So check this out. It loops around and inserts into the replisome like this. So this is that picking up and dropping process. So now that's inserted into the replosome can go the way that it's going to go. And as it synthesizes, it will synthesize DNA in the only way DNA polymerase knows how, right? From that 3' hydroxyl in the 5' to 3' direction until it hits the end of the next Okazaki fragment. It will then drop it. Right? And then the next time a primer comes around, it will loop around in the same exact way. So I highly encourage you to build your own model. It really, for me, was finally the thing that allowed me to understand this concept. So get those models built. Man, I could use another cool replication model. And of course, as is always the case, models are always worth extra credit. This does end our coverage of replication, so we're going to pause here and we're going to head next into the lecture on transcription. So the process of converting DNA code into RNA. So get jazzed. Today we're going to turn our attention away from replication and the nuances that we've just studied there to talking about transcription, or the process by which DNA can get converted into RNA. This is going to be a wild ride, and I want to make a note before we jump into the process of transcription that there are some things that do unify replication, transcription, and translation. That is to say that they all occur in the same 3 stages. So remember, with replication, the first thing that happened was initiation, when the origin of replication served as the starting point for the binding of the replisomes in the beginning of the process. We called that initiation. This is also going to be the first step in transcription and the first step in translation as well. So steps 1, 2, and 3 will remain the same in all 3 processes. The second step after initiation is elongation. In elongation, the subunits that are added one at a time. So remember, in DNA synthesis, that was the dNTPs, the building blocks, that were added one right after the other. Now, the third step is always termination. And of course, in termination, in replication, that was when we saw, if I can get my spelling right here, that was when we saw TUS come into action and terminator utilization substance and the hitting of that termination site and the stopping of the process. In transcription, things are going to be the same. There'll be initiation, but we'll have a different start site, and we'll actually call it something called the promoter. We'll get elongation, and instead of building with dNTPs, we'll build with NTPs because we're making RNA and not DNA. And then finally, termination will occur at a different site that we're about to introduce. Let's jump in then to the process of transcription and the wild ride of making RNA from the DNA code. Before we can get really jazzed about this, we have to make our vocabulary up to snuff, and we have to talk about some of the terminology that always comes around with transcription. First of all, when we think about the 2 strands of DNA, there's the 5' to 3' strand and the 3' to 5' strands running anti-parallel to one another. Just by convention, these 2 strands have been given names. We always write the 5' to 3' on the top, and we always call that the plus strand or the coding strand. So coding, plus strand, or 5' to 3' strand, all names for this top strand. Now, this all is sort of relative, isn't it? Because if we turned upside down and stood on our heads for a while, right, inversion's good every once in a while, we would see this as the 5' to 3' strand. So really, this is all about how you orient yourself with respect to the gene that is getting read. And so that's where we need to turn our conversation now, is to look at the gene and the regions that flank the gene. But first, I need to make sure to note that the bottom strand, you might guess this, is called the minus strand. It's also called the template strand. And that is very fitting because, as it turns out, this is a strand that is going to be utilized to make the RNA. That is, it's going to be utilized as template for the synthesis of the RNA. So the bottom strand, or the template strand, or the minus strand, is the one that really is going to be in the action when synthesizing the RNA, because it's going to be the thing with which the new NTPs that are making the RNA get paired. So it is really what does allow for the synthesis of the RNA. Now that's an important thing to bear in mind. But now let's orient ourselves with respect to this gene and recognize that the start site is the very beginning of the gene, so transcription begins at the start site designated +1. Now there's an end site as well, because we know termination is the last step here, so there's a transcription termination site as well. Notice that to the left of the gene, we see this blue region, and this is super important. Just the way that the ORI-C did in replication, this site called the promoter is going to serve as the landing pad for all of the machinery that are needed in order to transcribe the gene. So we might think of this as a region where everything can assemble, getting ready to read the gene and convert it into the RNA code. So if we picture this happening, and even without a lot of knowledge, we can say, okay, well, all this stuff is going to land here at this region in blue, and it's going to read down the gene. And the 3' to 5' or template strand is going to serve to pair off with the newly made RNA. So the DNA code right here is going to pair with the newly made RNA. And based upon what's going on this 3' to 5' strand, that will dictate what NTPs are added next in the elongating strand of RNA. So you might start to get the feel that in fact, because the bottom strand is serving as the code to make the RNA, turns out the RNA looks exactly like the top strand of DNA. Only what is true? And I can visualize Keenan like raising his hand, so I'm virtually calling on you right now and saying that in fact we know that the uracil replaces the T, thymine, in RNA. So although the RNA is going to look just like the top or coding strand of DNA, it's going to have U's every place that we see a T. So just in case you were zoning out just then because, hey, it's a vodcast and I'm not there, not necessarily wanting to make eye contact with the vodcast. And so just in case, I'm going to say this for the second time of what will probably be about 5,552 times, and that is that the RNA looks exactly like the coding strand of DNA. This one here looks exactly like the RNA, only in the RNA, U's replace T's. Now, Alex, he's out there and he's like, Rachel, you're oversimplifying because we recognize that in fact it is NTPs that are used to make RNA and not dNTPs. So not only does this differ in that U's replace T's, but we also must recognize that it is NTPs that build this that have that 2' hydroxyl that was not ever found in DNA, thus making RNA a much less stable molecule than DNA. So that's something to keep in mind as well that differs in the RNA. But sequence-wise, if we know the sequence of the coding strand of DNA, we know the sequence of the RNA, assuming that we also know where that +1 transcription start site is and what is the first nucleotide in the coding strand of DNA that begins at that point. Excellent. I promise I will say that again, but we're not done yet with lingo. We need to introduce a bit more vocabulary, one of which I have already stated, and that is the promoter. The promoter is this region to the left of the gene where all of the machinery assembles to begin the transcription process. It's like a landing pad or a start site for everything that's going to take place. Now, in the bacterial world, we recognize that sometimes there is more than one gene that gets controlled by the same promoter. And I'm actually going to take a moment to draw this because it's worth our time. If we have our top coding strand of DNA and our bottom template strand, and we recognize the location of the promoter to the left of the gene. So here I'm going to label it P for promoter. And then downstream we might have more than one gene. So let's say we might have gene 1 and then maybe gene 2 and then finally gene 3, all controlled by the same darn promoter. So what happens then is when RNA gets made from this DNA, we get the formation of an RNA. Of course, it's going to look like the coding strand, right? It'll look just like the coding strand, only U's will replace T's. And we'll recognize this to be the 5' to 3' orientation on the RNA. But this RNA will be special because it will carry the information for gene 1, gene 2, and gene 3. We have some special names for these things. DNA, in which a single promoter controls more than one gene, is called an operon. So this would be an operon. An RNA, an mRNA that carries the information for more than one gene is called, and you guys could guess this, what is that prefix for many, right? Poly. And then we have more than one gene, so this is polycistronic mRNA. Let's jot that down. So in bacteria, more than one gene may be under control of the same promoter. This is called an operon. Many operons are famous, and in fact I'm betting that Olivia is thinking right now, the lac operon. The lac operon is a famous one, and that's where more than one gene is under control of the same initiation site or promoter. Then we know that the RNA that's formed from an operon is called a polycistronic mRNA. It carries the information for more than one gene, often many genes, so polycistronic. In eukaryotes, we don't tend to see this. In fact, generally in eukaryotes, every single gene has its own promoter. And so we call that monocistronic. So monocistronic mRNA, a single gene under control of a single promoter, very different than a polycistronic mRNA.